I'd like to introduce Dr. Nicole Calakos. She's a professor of neurology and cell biology here at Duke, and she's the Lincoln Financial Group Distinguished Professor of Neurobiology, and she's done training at Stanford and also at UCSF.
Great. Thank you, Julie. It's great to be here. You have a really fabulous lineup of talks today, where you're going to hear a lot more in the afternoon about a really important mainstay and what everybody is doing to really make quality of life and improve that the best, as well as thinking about, can we do better? And this is kind of the question that captured and hooked our attention, which is, can we do better than the botulinum neurotoxins for dystonia? And we heard earlier that blepharospasm doesn't go away, there's no cure, and then Dr. Berman recently just told us it originates in the brain. It's not a muscle disease. So it really strikes me as an injustice. We're paralyzing the messenger and we're not getting at the root cause of the problem. And this is sort of why this is another neurologic disorder, restless leg syndrome. And the scientist is in there with the mice and cut off their legs and said, okay, we did it. Here we go. We fixed restless legs. So can we do better? That's sort of the question that attracted me as a movement disorder neurologist. And ultimately, could we get at the root cause? Could we do rational mechanism-based therapeutic strategies and get better treatments, maybe even a cure? But there are some very big challenges. Two biggies: what are the cellular mechanisms driving this? And what are the cells and circuits? So Dr. Berman and Dr. Bakari are going to be talking about what do we know about the neural circuitry in the brain causing this. And I'm going to give you some insights that we're having recently about the cellular causes and how that understanding is identifying new drug treatments. So when we talk about cellular mechanisms for dystonias in general, we do know some, but really not many. And the easiest one to point out is dopamine deficiency. There's a gene that's required for dopamine to make dopamine, and if you don't have that gene, you get a dystonia. You saw tardive dystonia earlier. That's taking a drug that blocks dopamine receptors and causes a dystonia. And then you also saw an example of it showing up in Parkinson's disease. So those are all examples of dopamine deficiency, but many of you realize taking dopamine or Sinemet does not help pretty much most of the dystonias. So where can we get clues to find causes? We do this by looking at the 10%, the very rare inherited causes of dystonia. And these are mechanistic clues to help us understand the majority, the 90%. And we call this sporadic or idiopathic, and I think idiopathic because we're still idiots and we have not figured this out yet. So let's get going. About a decade ago, we started with the one at the very top, DYT1. It was the first gene, and it was our first hook in. This is a dystonia that starts in childhood, often starts in the foot, and progresses up to involve the whole body. You heard from earlier about how the progression of blepharospasm sometimes. In these children, it'll involve the whole body. So what happened in our research was that we got a roadmap. We found that multiple genetic causes of dystonia and a subset of people with sporadic dystonia, this was the neck cervical dystonia in this case, were coalescing in a biochemical process that had been well described. This is used in every cell of your body to respond to stressors, to cell stress. We also heard about precipitants of serious emotional stress. Fundamental pathway in the body. Interestingly, in the brain, it moonlights and has another role in synaptic plasticity. in particular in the basal ganglia circuitry. So synaptic plasticity is what our circuits do in response to experience. They're important to cause long-lasting changes, like remembering our grandmother's cooking, our address, learning and memory, and also for learning motor movement. So they can be very long-lasting, but perhaps the disruption of the function of this pathway made that process a little extreme and on the pathological side. So in this pathway, the other key factor was that all three of the dystonias were causing sort of the same problem. They were blunting, if you activated this pathway, they were blunting it and weakening it. So this predicted if we boosted it up, activated this pathway, which I call the ISR for short, it's the integrated stress response, would that help? So we had a laboratory mouse model that had the human mutation for DYT1. And, oh, sorry, mice, there we go. And so far in these experiments, everything we give the ISR boosting drugs to have normalized. Cholinergic neuron hyperexcitability is a feature of many dystonias, and anticholinergics work in the clinic. Synaptic plasticity that we can measure is normalized. And when we gave the drug in vivo for a few weeks and then stopped, we had a long-lasting correction of white matter abnormalities on the MRI. That this was very helpful for us because both the mice and people have these MRI abnormalities. So that was a glimmer that maybe we could have long-lasting correction from intervention and treatment. The pathway is involved in making proteins, and so we thought we might also be able to see indicators of what state the pathway was in by looking at secreted proteins, and indeed those are abnormal and we can see corrective factors there as well. So this brings us to the question of what about blepharospasm? What we're seeing is growing evidence from multiple laboratories across the world that there are other genes causative for dystonia that coalesce in this pathway. And what this very busy figure does in the pink circles are genes for conditions that include dystonia. And many of these interact with this pathway and could be that origin. We have yet to understand, all those are rare genetic forms, are there intersections with what many people have for their dystonias, an unknown cause? So what we're trying to do here is see if we could look in the blood and have a biomarker test to see what kind of dystonia do you have. And this is the very first proof of concept using that DYT1 mouse. And all you have to see here is that these two color patterns look different. Each row is one individual mouse, and each kind of little bar is a particular protein. If they're red, there's more of them than in the disease model, and if they're blue, there's less of them. All you need to do is see the pattern. If we took the disease model and treated it with the ISR boosting drug, what would happen to these biomarkers? What would happen to this protein profile? And we see it looks more normal than abnormal. And this is really exciting to us. If we could have a way to identify ISR, people with dystonia due to ISR mechanisms, and a way to intervene. This is an HIV drug, ritonavir. So it's very interesting for repurposing. And there's also even better compounds we're developing. So the stage of the work right now is asking, does this translate to people? And this is a 3-site study to enroll with just a blood draw. And this is already preliminary data from the DYT1 people. Up here is just that the proteins are significantly different from healthy volunteers. So we're seeing signal, and we're using what we know to identify key biomarkers and move towards this. This is really important because with that tool, we might be able to ask the question, are there subsets of people with other dystonias that don't have those rare single gene causes that are mechanistic related? This can help us if we do get drugs into clinic to get the right drug into the right patient. We're really excited about that. If anybody's interested, move disres at dm.duke.edu or that QR code for the biomarker study, which is a single blood draw. And with that, I want to thank you from our group. We're very committed at Duke University in general. We have alliances across so many departments, as you're hearing today, and to give excellent care to people like you with dystonia. Thank you.